First of all, good evening and welcome to our third Mary Talk of our 8th season. My name is Philip Duggins and I'm with the Alumni Engagement Office here at Mary Washington. And it is my pleasure to welcome you to tonight's event. We are excited to host a sizable audience for tonight's presentation, which includes alumni, students, parents, and friends from across the country and beyond. As with past lectures, there will be a time after the presentation for our guests to answer any questions you might have. Feel free to ask questions by clicking on the Q&A icon below and submitting there, or vote for other submitted questions to help them rise to the top of the list. At this time, I'd like to introduce Kathy Wotecki. Kathy graduated from Mary Washington in 1969 with a degree in biology and chemistry and has served in many roles, including as the USDA's first Undersecretary for Food Safety, Chief Scientist, and Global Director of Scientific and Regulatory Affairs for Mars. Kathy, thank you for joining us.
And that was Mars, the company, not Mars, the planet. Well, good evening, everyone, and welcome to this lecture. I can't tell you how excited I am to be here and to hear Dr. Reif's talk. We've really been blessed at Mary Washington over many, many decades to have a small but mighty department of chemistry, whose faculty are as amazing in the classroom as they are in the lab. And tonight, we're going to be hearing from one of them, Dr. Randy Reif. Dr. Reif earned his bachelor's degree not too far from us in Virginia at St. Mary's College of Maryland in 2006. And then he moved to Texas to get a PhD from Texas Tech University. He did what many, many scientists do, pursued some postdoctoral studies, first at the University of Cincinnati and then at the University of Kentucky, before returning to his alma mater, where he was a visiting assistant professor. And we were able to lure him to Mary Washington in 2014. And Dr. Reif is currently an associate professor in the Department of Chemistry and Physics, where he teaches biochemistry, which is my favorite course of all time at Mary Washington, chemical analysis, and general chemistry. He'll be sharing with us tonight some of his research that relates to killing cancer cells. And with that as an introduction, I'd like to turn it over, Dr. Reif, to you.
Thank you, Kathy, for that amazing introduction. And thank you, Philip, for hosting this and inviting me and all the alumni board people for having me talk to you all tonight. I just learned about Mary Talks, and I think that they're pretty fabulous. And I am very excited to have the honor of presenting one. Now, I want to start by saying that first and foremost, I want to apologize now if you have trouble understanding me. I do get very excited when I talk about my research. And 1 year ago, about a year and 20 days to be exact, I had a stroke. I suffered a stroke, so I have seen the inside from the patient perspective of recovery and long stays in hospitals, and I've worked with therapists. And so many of those students that I work with in biochemistry, and I teach biochemistry too, I know now from the patient side anyway, what they're going into and what they'll have to deal with. They'll have to deal with crazy patients like me. And that's pretty cool. So that's one reason I note the stroke is just, hopefully you can understand me well enough. And I will try to speak as slowly as possible. The other reason I note it is because I was in fact prescribed during my recovery with a PPI. I took a prescription strength version of Prevacid, and I am now in love with these. It has been amazing what it has done for me. And my stomach, it is pretty darn fabulous. So I have personal experience with PBIs, as well as, of course, using them in my lab, which, yes, we had started using them in my lab to kill cancer cells, or trying to kill cancer cells anyway, before my stroke. And so it's been lots of fun now having that personal connection with PPI drugs. So, yep. So now, as a professor, I would like to say that a professor is nothing without their students. And so this is basically, these are two pictures that I have of students that have worked in my lab that have started this project. I'd like to point out Ash. Ash is currently in a PhD program at NC State, crushing it. And Shreya had been working on this project until she graduated in May of last year. And so these two students really started this project. And now I have current students who I don't have a photo of. So I'm going to have to get that ASAP. That's pretty embarrassing, who are working on this as we speak. And so I'm going to tell you a little bit about what we do. So I have always been interested in cancer. My graduate study days, all the way back, I got my PhD studying apoptosis, or programmed cell death, and in cancer cells. And so I've continued that here at Mary Washington as a professor. And so when it comes to fighting cancer, there are several tools that many of us know about. And so you've probably heard of chemotherapeutics, right? And these are the drugs, and there are lots of them. And they're known for attacking dividing cells, rapidly growing cells like cancer. And there is a wide expansion of treatment for cancer. Over the last 100 years, it has been of particular interest, and there has been a lot of research done on cancer, how to treat different types of cancer, how to kill those cells. And I am just joining the fray, if you will. And so we are always looking for new chemotherapeutic options, right? And so that is part of what has inspired a lot of my research. And so this has resulted in recent years of an increased prognosis, right, for several types of cancer. There are many cancers out there now that are not necessarily going to be the death of you if you're diagnosed with them. We can treat a lot of cancers. We can't treat them all. And that is still an area of interest, and it's still something we're working on. But it provides motivation to scientists like me to figure out how we can kill all the cancer cells, all types of cancer, and can we cure all the types that are out there. And so I am interested in cell death. Now, I am particularly interested in apoptosis rather than just blowing up a cell, or what we would call a necrosis. So when you have a healthy cell, there are multiple ways that there are really two major ways that it can die. It can die by necrosis, like where it blows up, its membrane is compromised. That's actually bad for us, for killing cancer anyway, because the route that I want to take, I don't want to activate swelling. I don't want inflammation. I don't want the body's immune system to be triggered. I want a process of death that is, for lack of a better word, natural to the body already. And apoptosis is that. So I want the cells, I'm interested in our cancer cells dying by apoptosis, not necrosis. That's the goal. And so when I say apoptosis, what I really mean is, I mean programmed cell death. So it turns out that all of the cells in our body, our cells are supposed to respond to stimuli and die when commanded to do so. And it's actually one very natural process. And it is one that our body relies on to maintain our size and our shape, right? While our cells are still dividing, we aren't getting much bigger, right? And that's because of apoptosis. And so apoptosis can be triggered in multiple ways, and we don't need to go into all of that, but I can talk about that. You can have a DNA damage response, or you can be signaled. You can have a signal to the cell, but there's a process for these cells that they go through in apoptosis. And that process is one that there are different tests that we can use to see whether a cell is undergoing apoptosis, depending on the stage of apoptosis it looks at. But the reason we love it, and the reason I love it, is because it doesn't leave waste. Apoptosis actually allows for the cell to be broken down in a controlled way, and the body can then reuse the broken down cell parts, right? So it turns out that macrophages will absorb our apoptotic cell bodies, or we call them blebs, right? And then we can reuse those resources, if you will, right? And that are broken down. And so when I say it's regulatory, it's happening all the time. One of the classic examples that I happen to have pictures from mouse embryos of is actually during fetal development. Now, this is a mouse, but it turns out that our fetuses do the same thing. We would actually have webbed fingers when we first develop, and it is apoptosis, these red dots here, that tells which cells to die where, and you can see the development on this mouse ball in just one day. You can see the cells, the area around the fingers there shrinking, right? And so it is a regulatory process that all of our cells should be able to undergo by themselves. Many cancers, as it turns out, tend to resist apoptosis or some of the signals for apoptosis. And that's part of why we get things like cancers and tumors. So, okay, I've talked a lot so far about apoptosis and cell death. I'm sure many of you are thinking, Reif, where are the heartburn medications? You know, you talked about these PPI things, these heartburn meds, where are they? Well, so heartburn meds that we are interested in specifically proton pump inhibitors, or PPIs, right? Things like molecules like Prevacid, right? Which is actually, its chemical name is lansoprazole, right? It's on the back, it's very fun. But Prevacid is just one of many out there available to us. And they're actually classically used, right, to help our stomachs when we eat this. Oh, I love this picture of chicken wings. I love chicken wings. I love buffalo. I love ranch too. What can I say? Living in Texas, Texas was all about the ranch with its buffalo sauce, ranch. So good. But it might give me heartburn. It certainly would have back in the day, right? And so you might be told to take by your doctor or by a medical professional, hopefully, or maybe not, but we sell them over the counter, Nexium and Prilosec, Prevacid. There are lots of options. These are all PPIs. These are all drugs that basically they're used because they'll reduce the amount of acid in your stomach. And so they're effectively antacid pills, if you will, but they're better than just antacids. Their jobs to work over time. So you take them usually on a regular basis. So why are we interested in them? Well, because one, they are extremely common in the US. You probably have heard and seen the commercials for Prilosec and Nexium. They are classically prescribed to treat gastric conditions. Absolutely. It's what the FDA has approved them for. It is what they do, right? Including my own. If you have an ulcer, for instance, your doctor may prescribe one for you. And they'll give you a higher strength, a higher dosage. Like I actually took Prevacid, but it was prescription strength. So the doctor put me on lansoprazole. And they basically said, okay, you're going to take this lansoprazole at a higher dosage than you can get over the counter. That was a medical professional prescribing that. It was pretty awesome. And it revolutionized my stomach. It is fabulous. When we dug into them, what they actually do is they inhibit what's called VATPase pathways. And VATPase is a proton pump. And it is actually what pumps acid into your stomach. And so when you take these drugs, Their job is to stop them from working, so they'll pump, well, slow down how much they work. Doesn't stop them, thank gosh, right? But it'll slow them down, inhibits them, so that you don't get quite as much acid buildup in your stomach, and you're less likely to get that heartburn and feel that pain, right? That you would get from eating those wonderful buffalo chicken wings that I love so much. Now, they're also used, it turns out, they are used as a supportive therapy in cancer patients already for their jobs. Like I had an ulcer when I had my stroke, was in the hospital, but many therapeutics will, in fact, these chemotherapeutics that they give to cancer patients, they will cause all kinds of side effects. And I'm sure you've heard about chemotherapeutics and different chemotherapies and different drugs that cause different issues. So one of the PPIs, one of the uses for proton pump inhibitors now is to help out the stomachs, basically, right, and the digestive issues and regulate the digestive system for cancer patients with chemotherapeutics, depending on which ones they're taking and what those effects are. They also, PPIs are fairly safe. They really are. Their adverse effects are extremely rare. And so they're generally considered very safe from the FDA. And that's why they're sold in multiple forms over the counter. Now, my interest is in using them to kill cancer cells. Right now, I am doing all of this, by the way, in a test tube, not in the human body yet, maybe one day in the far, far future. But we kill cancer cells that we grow ourselves. And I'll talk a little bit about that in a second. So, why PPIs and why are they interested in killing? How would they possibly kill cancer cells? So, it turns out that many cancer cells, not all, but many cancer tumors, they actually have these proton pumps, and they have lots of proton pumps. They make a lot of them. And so over here, we see the inside and outside of a cell. And this, on the right side, is a tumor. It turns out that many cancer cells will create and use extra proton pumps to regulate the fact that they are metabolizing a lot of extra stuff. They're doing a lot of metabolism. They're burning a lot of energy. And so they're going to pump out the acid, if you will. No, don't spoil it. They're going to pump out that acid out of the cell into the outside of the tumor, right? And so that's part of their growth. That's how the cancer cells grow, and they grow quickly, and they grow up, and they become these masses. And so what we'll actually see, what we know, is we've noticed that the environment around cancer cells is fairly acidified because of these proton pumps. So our hypothesis is that if we were to stop the proton pumps from working, the cancer cells would not be able to regulate all of these processes, and they would likely not be able to control the acid inside them, and they would hopefully die. Hopefully, extra hopefully there, by apoptosis. So the PPIs that we have studied in my lab and we've used we've looked at 3 of them. And these are 3 PPIs that are available that we can get through chemical manufacturing companies directly, rather than having to buy them. We don't have to buy those pills, for instance. Now, I know I apologize now for the chemical structures. I geek out of them. I know that not all of you will. It's okay if you don't. We won't talk a whole lot about the chemical structures here, but I will point out, because it's really fun, that Prilosec and Nexium are actually very, very similar in structure. To put it simple, Prilosec is a mixture of omeprazole. They call it omeprazole. It's actually a mixture of 2 hands, right? And then esomeprazole, sorry. Nexium is really just one of the hands. They're very, very similar. And then we use dexlansoprazole. Dexlansoprazole is only available by prescription, or you can buy it from chemical companies like I do to use it on cancer cells. You'll notice that it does in blue here have a very different chemical structure. Similar base, different structure. And that's all that matters. That's all the chemical structures I'll talk about. Don't worry. And yes, I do include the abbreviations that I'll use later in the talk when we get to the data portion. So these are the 3 PPIs that we could basically get our hands on for a reasonable price to test with our cancer cells. So the research goals for my group, and this started a couple years ago with ASH, was to basically use PPIs to induce cancer cells. And we want to answer several questions, really 2 really big questions. So the first one is, do PPIs kill cancer cells? And the second one is not just do they die, but do they die by apoptosis? And so that is what my research group and the students working with me, we've been working on for the last really now 2.5 years, which is pretty awesome. So how do we do this? So we do this using microscopy. And so this is it's light microscopy. We're going to make our cells, we're going to make them glow, and we're going to literally look at them with our eyes. It's pretty cool. So we're going to use microscopy to do that. And we're going to make them grow. And I will talk about how I'll make them glow. We'll make them glow. And we'll visually inspect them with a really cool microscope. And we have one similar to this. This isn't our exact microscope in the Jepson Science Center, by the way. If you ever want to see it, if anyone happens to come by, feel free to let me know, and I will take you to the microscope room, and I will show you our beautiful microscope that I geek out. I just love talking about. I could talk all day about the greatness of fluorescence microscopy. So there it is. That's how we're going to do this. Well, okay, so we have an instrument we're going to use. We've talked about these cancer cells. What are the cancer cells? So the cancer cells that we study, we're going to study an immortalized cell line called the Jurkat T lymphocytes. These are actually white blood cells. These are T cells that are cancerous. They're immortalized, meaning that they will grow indefinitely as long as they are given the nutrients to grow. And they actually grow in flasks like over here. And so they look like this. They grow in solution, right? So they'll grow in a special solution with all kinds of nutrients and food for them. And they'll grow as long as they have food and as long as you keep their environment at the right pH, and you give them the right amount of air. It's very exciting stuff, and they'll keep growing, right? And they are the gold standard for apoptosis studies. And part of the reason why is because, one, they're easy to culture, so they're easy to grow in the lab. And so students that go through my lab, all those students we saw pictured before and more, are generally trained to grow the cancer cells on their own. I teach them how to handle them, how to grow them. And so doing cell culture is part of what I teach them here at Mary Washington. Because these are particularly easy to grow. They also grow quickly. They grow pretty fast. Really happy Jurkat cells, when they're really happy and when we've kept them in the right conditions, they'll double in about 24 hours, which is pretty darn awesome, right? So that's great. They can grow fast and they're easy to grow. Easy is, of course, relative, but I train the students. That's great. And the third reason they're the gold standard is because they are susceptible to apoptosis. They're actually known to be susceptible to all, well, not all, but most of the pathways, the 2 major pathways of apoptosis and cell death, they are known to be susceptible to both. Many cancer cells are not susceptible to both. They resist one or the other, whether it's a signal or damage. They resist that. That's why a lot of the reason why they can become cancer. And so Jurkat cells, turns out, they don't resist that, and they grow fast enough that we can use them. Now, apoptosis, you can actually see signs of apoptosis on the microscope. So these are some images that my student took a few years ago, showing that over time, you can see a cell here highlighted in yellow, circled. We zoomed in on it, and you see that over the span of a couple hours, they'll also go through the apoptotic pathway. They'll go through apoptosis pretty quickly. And so in the few hours they saw, in about the 3-hour time span, they went from a cell that looked healthy. It wasn't. It was undergoing apoptosis. We just couldn't see it morphologically yet. We couldn't actually see the blebbing. but then it's blebbing later. And you can actually see these little apoptotic bodies and blebs all around the cell as the process kind of reaches its conclusion. So it's very exciting. That's the other reason we love Jurkat cells. They also undergo apoptosis fairly quickly as well. So for our experiment, what we're going to do, the experiment that I'm going to present to you today with some data, with some awesome pictures, the basic experiment is going to take cells. So it starts with cells that we grow in a flask. We would put them in a tube, and then we're going to move them to a petri dish. And we're going to put them in this pink solution that has the nutrients they need. And we're going to add to that the PPI drug, right? And we're going to add a probe. Now, the probe can absolutely change depending on what it is we're looking for. And what I'm going to talk to you today about is actually just death. We're going to look for a probe that tells us whether cells are alive or not. And so what we are going to do in the experiment I'm going to talk to you about is we're going to take our cells, and so we have our petri dish, and we're going to take some out, we're going to put it in a tube, and we're going to split our cells into two sets. Half of our cells are going to get no stain to make sure that we can see that we can check on the microscope to see that we're looking at color, where it's actually going to show us whether they're alive or not. And then the other set we're going to stain with calcium AM. Now, calcium AM, the name is not important. It is simply a probe that will make healthy cells glow green. And so we're going to take images in both white and green light, and we're going to look for green glowing cells. Green glowing cells are cells that are alive. And so that's really exciting. And so we have our first data slide. Now, this, I know it's a lot of pictures, but let's just break it down. So what we're going to do is we looked over a span of time. And so this is a picture at 0 hours, 18 hours later, and 30 hours after that, 30 hours total, right? After exposure to the drug. Now, we have 3 sets of images here on this slide, and I have more to show you, don't worry. One of no drug, where there's no drug, so these cells should be healthy and happy all along for all the time, and we can see that there are lots of green dots. Lots of cells are glowing green, showing us that, yeah, these cells, they're loving life. They're very healthy. Now, doxorubicin is a known chemotherapeutic agent. It is a known apoptosis inducer. And so we use that as a positive control. So we know that it should kill the cells, especially by hour 30. And we expect that the cells will die. And though they should die by apoptosis. And that's why we see lots of green dots and then fewer green dots and far fewer, much less green by the time we get to 30 hours. Then we're going to compare that to our 3 PPI drugs. The first one is the omeprazole. So we do that first, and we see that omeprazole, yeah, there's a little bit of a decrease in the green cells. Dexlansoprazole, there's a pretty dramatic decrease that we can see just by looking for the green dots. Esomeprazole, not so much. And here's omeprazole again. We put it on for comparison with the different PPIs. Now, these are just the raw images. So my 4 students in my research group, so one of the things we would do is they got very good at taking these images and counting the cells and calculating what percentage of the cells were alive and what percentage were dead. So they made a graph. So here we go. We made a graph of percent of cells alive. And so the 100% represents the fully alive, right? And then at 0, all of them. So we have our 4 drugs and our control. And so you'll notice that consistently the no drug blue line at the top is consistently the highest, as it should be. There's no drug there, right? And what we see that is really interesting to us anyway, the most interesting part we've seen, is the fact that over the span of 30 hours, the dexlansoprazole, right, seems to kill the cells pretty dramatically, right, especially relative to the others. When we compare at 30 hours, numbers-wise, those percentages, the raw percentage, yes, the nextland result is statistically different. It is statistically more dead than the other drugs. We compare the drugs to each other, right? And so that is pretty darn promising for us. We are very excited about that. Exploring the nextland result, right? So the takeaway message is, for right now, do the PPIs kill cancer cells? Our data suggests absolutely the dexlansoprazole is killing them, right? Do they die by apoptosis? Well, that has been a debate. We are still doing more studies. That is still unknown. We don't have clear data on that. But what we are currently studying is actually related to this image that we go back to in the dexlansoprazole. So my current students, Emma and Caroline, are working in my lab to see. They have dramatically, they noticed, and you may have noticed it too when you look at these images. What you notice is that there are less overall cells there. It turns out that when you look in the white light, that is true as well. There are less overall cells. So they're actually trying to answer an additional question, which is are the PPI drugs also slowing down the growth? So the dexlansoprazole showed death. So we killed cells that were there. Is it also preventing the cells from growing? And that is a current question that we are trying to answer in my lab as we speak. And unfortunately, I apologize. We were literally like about a week or two off. They have, my students have the pictures. They are counting the cells right now. And I didn't have time to incorporate it into this presentation for you. But they're counting up the cells as we speak. So we don't know how it turned out, but had this presentation been a week or two later, we'd have had even more data to show you, which would be really exciting. And so that's where I'm going to leave this off. I could talk a lot more. Anyone who knows me knows Reif never stops talking. And so I'm going to stop myself by just acknowledging and thanking some really important people. The research group that has done work related to this, that I'd like to thank. My current students are Emma and Caroline. I'd like to thank Ashley and Shreya. They actually started this project. Ash actually was reading papers and noticed that there was a paper that talked about how omeprazole induced apoptosis in Jurkat cells, but didn't explore it. It just noted it. And so if it wasn't for Ashley bringing this to me, I would have never noticed that. So Ash brought that to me. It was awesome. And so that's how this started. And Shreya continued that work, and Emma and Caroline are continuing the work now, which is really cool. And so I'd like to thank all of my group members that have been doing the apoptosis and the cell work. They have been a fabulous team. Yes, I graduated, a bunch of them gone off to do awesome things. I could talk forever about where they went. It's so exciting. I would, however, like to thank a couple entities a lot for funding this research and for providing the opportunity to do it. I'd like to thank the Summer Science Institute at University of Mary Washington. Several of these students have done summer science with me over the summer. I'm working on this project. I'd like to thank the departments of chemistry and physics, as well as biological sciences. The microscope is absolutely a collaboration between myself in the Department of Chemistry and Physics and the biology department. It has been really exciting. I'm so glad that we have an awesome light fluorescence microscope. It has been great. I would also like to thank Irene Pisco-Rogers and James Rogers for their student research fellowships that they created. They have funded. That has funded several of these students, including Emma now, is actually receiving some of that funding for this work. As well as, of course, undergraduate research grants that we can get from UMW that they have given us to allow us to buy things like these drugs and to buy the cells and the medium to keep them alive and to grow them. All of that costs money, of course. And so I'd like to thank the university and its various funding sources for that. And I would like to thank the Virginia Academy of Sciences for providing a grant most recently to Shreya, who they funded. They gave us some money and we were able to buy more drugs to use in this project. So with that, oh yeah, we're excited, of course, and it's really, really fun science stuff. With that, I'm going to finally stop talking and take questions, and I'll talk about anything you want to talk about. Then I hopefully that I maybe know about, and if I don't know about it, I'll tell you honestly that I don't know. But thank you all so much for listening. It has been super awesome.
Great. Okay, great. So just as a reminder, if you do have a question, please use the Q&A button at the bottom of the screen, and you can obviously see the questions that have been submitted so far, and you can upvote if you see one that you specifically want to have go to the top of the list. But we'll start right now with the first one, and someone that you just mentioned on your slide, Kat Zwimmer, is actually says, "I love you, Dr. Reif." So just that's the first one. Then the next
Thank you, Kat.
the next one is from Henry, and it's, "How about pantoprazole? I take it regularly for my ulcer."
So pantoprazole was in fact also prescribed to me during my stroke. We don't study it because we cannot get a chemical company to sell it to us right now. I'm assuming it's probably because of something related to patents and production. And so we are not able to get pantoprazole. Yes, pantoprazole is in fact another PPI. And I would love to get my hands on some of it for laboratory use. It would be amazing. Yes, I also was prescribed pantoprazole for my ulcer. And yeah, it's pretty amazing stuff. It helps treat that. So yeah, no, pantoprazole would be great, but unfortunately, these are the three we can get right now. We would love to expand this. So, if anyone hears of companies selling to laboratories to use, of course, I would love to know that. Pantoprazole, absolutely. I would love to study pantoprazole. Unfortunately, nope, we can't get our hands on it. But I've had my hands on it, and it helped me a lot. I love that stuff.
All right, next question from Jennifer. "This is fascinating. Have you gotten grants for this work and/or working with any cancer researchers?" And I know you mentioned a few of those, but if you want to go into more detail.
Sure. The grants we have received so far for this have been the only external grant we've received outside of UMW is the Virginia Academy of Sciences. So the short answer is yes. They give small research grants to undergraduate students like Shreya, like Emma, who go for them. And I will be having Emma write for one, write for another one next year. They're generally between $500 and $1,000. And so, but every little bit helps a ton. But so the Virginia Academy of Sciences has provided grants for this. Right now, we're still gathering the preliminary research enough to go for larger grants. We're not quite there yet.
All right, next question. "Regarding the cancer cell line that you are using, what type or types of cancer, from a layperson's perspective, would they typically cause?"
Oh, so this is a leukemia cell line specifically. So the Jurkat, so I'll go back to the slide about Jurkat T lymphocytes. They actually they obtained these cells from a 14-year-old who was suffering from leukemia. And so these are it's a leukemia, so it's a blood cell line. And that's why it's T cells. Now, it turns out that the reason we use them, I talked about in detail. The other reason we are fortunate enough that Jurkat cells do rely on that VATPase, that particular proton pump, that the proton pump inhibitors stop from working. They inhibit it. And so right now, that is all that I can confirm, is that it's working with leukemia cells currently. We would love to explore other cell lines, and does it do the same thing? Once we know how it behaves here, then we could try other cell lines in the future. Absolutely.
Next question. "Do you hypothesize that results will be consistent over various types of cancers and/or stages of advancement of cancer?"
Oh, Do I hypothesize? So, okay, you gave me permission to make stuff up. So based on my knowledge, I would say it is unlikely that these drugs are not very likely to work on all cancers, all types of cancer. No. Now, over the stages, based on my understanding, the stages are really based on how far along the tumor growth has gotten and whether the tumor has spread. So I will admit that I am not a medical doctor. And so I'm anticipating, though, I would anticipate that if we're able to kill cancer cells and we're able to kill that type of cancer, say a T cell lymphoma, right? Yeah, if we're able to kill it in the lab now, would we be able to kill it depending on the stage? Probably yes, because it's really going to be dependent on whether the cancer cells, do they rely on those proton pumps to maintain their internal environment. And so the short answer is, yeah, I would anticipate that depending on, really should be independent of stage. I would be surprised if the stage of the cancer affected that. It's really going to depend on, does the type of cancer doesn't create and use those proton pumps that our proton pump inhibitors are stopping from working. And that will absolutely vary based on type. That is very true.
All right, Sierra asks, I love this research. My question is, by using PPIs to induce apoptosis, how would the autophagy affect the time of growth in cancer cells?
How would autophagy? Oh, okay. So so Sierra asks, okay, so in what we do, there are no macrophages basically to actually to suck up the parts. And so we're not going to have to worry about, and I can't get it to change for us. We're not going to have to worry so much about the immune system. There's no immune system there to worry about. It's basically, do they grow or not? So we are working under a very simplified model. But that being said, the question really would be for us, would be what's the best way, and can we confirm that apoptosis is really going in our cancer cells? And so far, we've had somewhat conflicting results. But because we've only been working in a tube, if you will, for lack of a better word, in a tube, in a culture bio in a petri dish, we don't have an entire immune system to worry about. We have one type of cells, and that's it. I hope that makes sense.
All right. Do you think that using PPIs on cell lines other than jerk would yield similar results?
I think that it would yield similar results depending on whether those cells created or relied on the proton pumps to survive. And so if we go to this slide, it would really be dependent on whether or not the proton pumps that are PPIs that we have, whether our PPIs will target those proton pumps. Now, there is a question, I was wondering if anyone's going to ask it, right? That goes along with that idea, with a common theme that I'm getting from the questions. That is really good. There is a question that I'm not sure if anybody's asked yet about basically how do we get the PPIs to the tumors, right? Would be a really important question. So not only does the tumor of those particular cancer cells, do they produce the proton pumps? One of the really interesting questions for the use of this far down the road, if we were going to use them as chemotherapeutics, we would have to address the fact that we can't just take a pill of them. The pills that we're taking, we're already taking pills. Those pills are being digested in our stomach. They're actually doing their action in the stomach. We would have to modify these in order to have them work. We would have to modify how they're delivered to the cancer, like maybe through the blood or some other way. And in doing that, the reason I point that out now in answering this question is because it would probably rely on a future step where we would modify the chemical structures. Where are my chemical structures? We're going to rely on having organic chemists modify. And then that would allow us to not only reach the cancer cells in different parts of the body, wherever they are, it would also allow us potentially to target particular cancer types, depending on what we end up seeing. We can actually adapt them to what kinds of proton pumps they use. It is possible to do that. That's going to be a lot of work in a very different research area. But it's far down the road, but very cool. Very cool.
Next question is, are the PPI manufacturers aware of the research you are conducting? Perhaps they can help with funding?
Short answer is no. The PPI manufacturers, so unfortunately we're fairly small relative to the large manufacturers of these drugs. They would probably make the argument that they are available through many of the chemical companies that produce the chemicals, like Fisher Scientific and Thermo Scientific and Sigma Aldrich. They're viable. They're purchasable. So many of them can be purchased. There are some that cannot. It would be interesting if a chemical company that's producing pantoprazole would be willing, for instance, something we can't buy like pantoprazole, whether they would be interesting to provide that. I have not explored that. That is a really interesting idea to explore whether or not they'd be able to make some for this use to try out. That's a really good thought and idea. I love that question. That's a really good one. You got me thinking. I like that.
Good. All right. Amy asks, are there any studies indicating that people who have been on PPIs long-term are not developing cancer at the same rate as the general population?
I don't know about that yet. So people haven't looked at answering that exact question. And that, unfortunately, that exact question is bigger than my research could answer. But my guess, based on what I've read, would say that really the method of action, what the PPIs are doing when you take them, they're unlikely to reach outside of your digestive system. So you put them in the digestive system, they're working in your stomach. It's unlikely they're going to reach other places in their whole form to inhibit cancer cell growth. But who knows? It is possible. No one has looked at that I know of. I have not read any studies. But if they do, that'd be great to know. That's a really good question. I wish I knew the answer.
Okay, next question, Renee asks, some cancers that metastasize to the bones create an acidic leaching environment in the bones, often treated with biphosphonate drugs like those that treat osteoporosis. Would PPIs do a better job?
Oh, I don't know. That's a fabulous, that is absolutely the method of action. That's how the PPIs would work. They are known to interact with the acidic environments, and they are known to basically, when they work better in an acidic environment, that's why they work so well in the stomach, and they would work in the area where cancer cells are actively growing. So I would say they work well. Would they be better than the chemotherapeutics that are known? We don't know. The short answer is, as of right now, probably not. With modifications in the future, quite possibly. But as of right now, I'm betting the known studied chemotherapeutic agents they're using for those particular cancers are probably still better. Okay. But that's a great question.
Next is more of a comment. I've always heard long-term use of PPIs can actually cause stomach problems because acid is needed to process food.
So again, as a non-medical doctor, I absolutely would not use them. That is, I actually have been warned. I can only speak from my experience. And so I was warned as a patient to make sure that they gave me the PPIs and they prescribed them, and they ran tests to make sure to wait until my ulcer was healed. And they too, the doctors did warn me of not staying on PPIs for very long, for long periods of time. And that is because absolutely I can imagine biochemically that you would not want to basically have for years and years having your stomach cells be exposed to them and have them inhibiting those proton pumps, because you do need your proton pumps to work in order to maintain your gastric juices at the right acidity level. And so I absolutely can believe that there is a finite amount of time that doctors would recommend you stay on them. Absolutely true. I know that was true with me. Absolutely. They tested it and they took me off. But the changes have stayed around. I have to say, my stomach has never been better. Just throwing it out there. I love PPIs. My stomach's amazing, and I'm off of them now. And I was on them for a solid 8 months. I'm off now. I love it.
Okay. Sue says, I had estrogen positive, HER2 negative breast cancer. Would the PPI kill those cells?
I don't know. We would love to test that. That would be a cell line. That would be absolutely, if we can get a cell line. The HER2 negative, yeah. I mean, we'd love to try that out. If we had an immortalized cell line or cell line we could grow in our lab, that would be that'd be a great question to answer. I don't know. As of right now, we don't know that yet. But that absolutely would be something we would love to try.
Okay. The next question, can you modify the chemical structure of the PPIs at your lab?
So that is such a great question. I am a biochemist and a bioanalytical chemist. So the short answer is me, myself, no. There are other chemists in my department like Dr. Oldham and Dr. Asper who love that organic stuff. They love these molecules. And I'm sure if Dr. Oldham were here right now, he'd be laughing hysterically at my awesome molecules here. And he'd be like offering, I could have them redrawn for you, make that look better. No, we would need an organic chemist to do those modifications. Is it possible to do it at UMW? Yes, actually. No, we absolutely could. It'd be a matter of figuring out what modifications we want to make and where on the molecule and why, and then having students also explore and do research on, can we make modifications to these? But the short answer is yes, we could do that. That is, again, and that's why I would categorize that as future work, because can I do that in my lab? No. At UMW, we have organic chemists that could, yes. Yes, they could do that.
Okay. Next question from Janet. If someone's taken one of these meds for a long time, will it help prevent any types of malignancies?
Also unknown. We don't know. The short answer, I mean, I would have a hard time. I would have a hard time believing that these drugs are making it outside of your digestive system as they are right now, the way they're being taken, which is typically as pills. And so I think that it's unlikely that they're going to be killing cancer. Although I'd love to be able to say that they prevent cancer growth. That'd be great, but no, we haven't seen the studies on that yet.
Do you think it's possible that people taking PPIs could unknowingly prevent cancer starting to grow within them?
It's possible. It is possible that unknowingly. It is possible. We don't know. Is it possible? Yeah. Do I think it likely? Probably not. Just because I suspect that once you take the pill, you put it in the acid, you put it in your stomach, it's going to get used pretty fast. It's going to start inhibiting the proton pumps that are there. It's not going to make it into your bloodstream or to where the other cancers are. But it's possible. I don't know. I'm guessing. That's only based on my experience and what I know about how they work. It's a great question. I can hope so.
Okay, next question. Do you have a prediction regarding how long someone would need to be taking PPIs to be cancer-free?
No. Short answer, no, I wish. Oh man, I wish. I'd be loaded, right? Oh man, that'd be great. That'd be such a great question to ask. Like, I'd love to be able to answer that. No, we don't. I mean, really, all we're doing right now is we're exploring: do they basically kill known cancer cells by apoptosis that we could grow in a lab? So I mean, we haven't been able to expand out yet to the other cells. We'd love to be able to do that. Other types of cancer, and then eventually, even can you imagine like human trials? Oh, that'd be incredible. But they would probably require, like I said before, it would probably require modification though, in order to make them work, to make them do that. I'm betting we could actually make them better killers of cancers, of other types of cancer, if we did some modifications. But I wish.
Okay, a couple of comments here to kind of wrap things up. I have been on PPIs for 35 years. I cannot be off of them because of a hiatal hernia. I'm okay, and my MD stepson said the consequences outweigh the risk, i.e., esophageal cancer. Who knows? Just putting that out there.
Yeah.
And then another question: do you know if stomach cancer uses protein pumps? Because that seems like a good place to start, since the drug could possibly be tested in the body prior to modification.
That's a really good. No, that's a really good. I haven't really thought. I would assume that they do. Stomach cancers would, but that's a really good question. That is a really good question. I don't know. That's another really great thought. Like, that's a great question, and yeah, it's definitely going to get me thinking. That's fabulous. I don't know. I would think that taking them would very likely would definitely influence the stomach cancer cells. The question is whether the stomach cancer cells have some way to resist and not care about them. And that would be a bit of a bummer. But I don't know. That's a really good question. That's a great idea.
And then there's one last comment here. Randall, your presentation was lucid and intriguing. I am so glad to see your recuperation is progressing nicely.
Thank you.
All right. So with that being said, we have reached the end of our time together tonight. So thank you, Randy, for joining us this evening. I'd also like to thank Mark Simpkins from Events AV, who helped facilitate the virtual logistics of tonight's program. And thanks again to our viewers at home for joining us and being a part of the conversation. Remember that this presentation will be available for viewing on the UMW YouTube channel by this time next week. There you can watch this program and browse the full catalog of Mary Talks. Our 8th season of Mary Talks will continue on April 17th and will feature Dr. Antonio Berenice, Professor of English, talking about Mary Washington's rare book collection with a talk titled Empires in Print, a Biblio Mystery from UMW's Rare Book Room. Keep an eye out for more information for our next presentation in March. We look forward to seeing you then, if not earlier. Thank you and good night.